Okay, today I want to focus on what happens to glucose and what are the different options, because we usually think of glucose and then glycolysis, and then glycolysis gives us pyruvate, and then that gives us the precursors for acetyl-CoA, which allows us to start the citric acid cycle. So that's kind of the direct line of glucose. That's usually the way that we think about it. But there's a couple different pathways that glucose can undergo in terms of chemical processes and metabolism. And I want to focus this little kind of mini lecture on vocabulary because a lot of the terms look very similar to each other. So I'm going to kind of couch it in a little concept map here. not this. It'll get bigger, I promise. But the vocab can get a little confusing. So I just wanted to kind of walk through it, and then hopefully that will help to clarify. Just because sometimes the language could be the tricky bit. You know, you're learning a new language when you're learning chemistry or biochemistry. On top of everything else. So let's just focus this one on the vocabulary. Okay, so we have glucose in our blood, and that gets there from chewing food. We're chewing food, breaking things down. We're getting these glucose molecules from our alpha amylase in our saliva and the various processes of our digestion, maybe from the pancreas, and getting absorbed in our bloodstream. It's coming in through whatever point that it's getting brought into our bodies. Now the glucose in our bloodstream can get pulled into cells, and as soon as it gets pulled into cells, it gets phosphorylated with glucose. So now it's glucose 6-phosphate, and that phosphate comes from an ATP. So an ATP becomes an ADP, giving this phosphate that's now on my glucose, on the 6th carbon there. So this is happening in cells, so in all cells. So now we have the glucose 6-phosphate in the cell, and now it can't get back out. So now it's going to do the things that you want it to do to get it to the other parts of the process, which for most of what we think about, again, most of what we focus on when you're first learning biochemistry as well, this glucose 6-phosphate now can undergo glycolysis, and the name of the game there is pyruvate. So glycolysis. So let's look at that term first. So glyco, of course, referring to kind of the glucose part. So anytime you see this prefix glyco, that's referring to glucose. And then lysis means to split apart. So we're breaking something apart, we're breaking it down. So we're splitting. Decomposing. So that's that 10-step process that goes from glucose to glucose 6-phosphate, to then the rest of the 9 steps to get you to pyruvate, and then pyruvate again can go to your acetyl-CoA, and then that goes into the citric acid cycle or the Krebs cycle. Okay, if we're kind of thinking about where this goes, and then of course citric acid cycle gets you into the electron transport chain, which gets you all that ATP, which gives you all the energy, all the things. Okay, so glycolysis here is kind of the vocab word. Now, we can actually go in the opposite direction. You can start with pyruvate, or if you end up with too much pyruvate, then you can go in the opposite direction. And you can reform glucose. You can form new glucose, and we call that gluco. Okay, so of glucose fame, neo means new, genesis, think matrix, genesis means formation of, so genesis like creation, neo is new, and then gluco in the same prefix, same with glyco is indicating glucose or anything that's kind of sweet in general. Gluco means new glucose also. So glycolysis in one direction, gluconeogenesis in the other direction. So we're forming new glucose. Okay, now the way that this happens is through some amino acids. These amino acids have to get involved here with our pyruvate because we're kind of undoing all of this stuff. So if we kind of think of kind of pillars alongside of our metabolism here, if we have lipids kind of coming off to the left, carbohydrates here in the middle, proteins off to the right, then the proteins that are kind of doing their thing over here, they can jump back in and kind of have an impact on this process along the way. We'll talk about that when we talk about protein metabolism in a future video. But for right now, those amino acids can kind of aid in this process, and this is happening kind of parallel in the concept map of my mind, but also parallel in the digestion that's actually happening in your body. Okay, now we can also go from glucose 6-phosphate back to glucose into the blood. So that only happens in specific tissues. So that can only happen in the liver. So in liver cells, we can go from glucose 6-phosphate, it's not irreversible, to the glucose in the blood. So if we end up kind of going through this process where we're doing the gluconeogenesis, now we've created this, if we have it in the liver, then that throws it back out into the blood, and then it can be reabsorbed in other places, wherever it's needed. Now this glucose in the blood comes from glycogen, like I said, so, or, other kind of large chain carbohydrates. So glycogen is a series of glucose molecules. It's a polysaccharide. All of these glucose molecules that are stuck together forming our glycogen, and that glycogen can form the glucose 6-phosphate, or the glucose 6-phosphate can react together to form the glycogen, because this is the way that these things bond together as well. So when these things bond together, we phosphorylate, then these glucoses stick together, and that builds up my large chain molecule here, my glycogen. It builds up my polysaccharide. So if we're building up or creating or forming, then now this is called glyco, again referring to glucose, or in this case glycogen. Genesis, formation of. So glycogenesis is once this thing is phosphorylated, then now we can stick these things together. So those phosphate groups kind of act as a way to stick together my monosaccharides to get me that glycogen. And then that breaking down of glycogen into the glucose 6-phosphate is the opposite of this. So if this is glycogenesis, the forming of, then we would guess we need some lysis into this one, so that we have glycogenesis. So glycogen, the term's right in there. Lysis means splitting of, right? Glyco or glycogen, right? Gen kind of is on both of them there. All right, how are we doing so far? Lots of arrows in there, that's pretty good. This is, I love a really complicated concept map, because this is kind of like a window into my brain most of the time. Okay, so let's review. Glucose in the blood can become glucose 6-phosphate, then that undergoes glycolysis, so we're splitting apart into pyruvate, which is the name of the game to get down to the citric acid cycle. Pyruvate, though, through some other factors, can undergo gluconeogenesis, which is the formation of that glucose 6-phosphate from neo, and then that can form glycogen through glycogenesis. That glycogen can be broken down between glycogenolysis, and then now we have our glucose 6-phosphate either way. And then we can kind of undergo the process. It could go that way directly, it could go in this way. Now the last option here is to kind of go off to the side, and this is usually just sort of a side note in a lot of introductory texts. You can get into it in a lot more gory detail if you go on in biochemistry, but this is called the pentose phosphate pathway. Pentose phosphate pathway, triple P. So pentose tells me it's a 5-carbon sugar. Phosphate, something to do with phosphate. And pathway, this is the way that we're getting there. So what we end up with is 5-carbon sugars and an NADPH. So NADPH is one thing we get out of this. And again, I'm not going to get into the gory detail of this process. They're still kind of working on understanding it, and research is being done at various levels for that. So I'm teaching at Central Washington University. One of the biology professors, for example, there studies this particular pathway in her studies of cell biology. So pentose phosphate pathway forms NADPH, and it also forms 5-carbon sugars. Sort of specifically, I'm just going to mention ribose 5-phosphate. Ribose 5-phosphate. and it'll give you pentoses in general, but I'm bringing up this one in particular because ribose 5-phosphate is part of the sugar phosphate backbone in DNA. So this is an important part of making up kind of nucleic acids, which are DNA and RNA. So this is kind of its own separate thing and its own separate importance, but it's another place that we can get this information. So the cool thing about carbohydrates is they're for energy. We talk about energy all the time because we think about this pathway, right, that we're getting all this ATP at the end. But we say that, you know, another important function they have is in structure. And so the structural component mostly is in, at least for animals, in creating these backbones for our nucleic acids, which are creating the backbones for DNA and RNA. So that would be something like the ribose 5-phosphate there. Okay. So we have glycolysis, gluconeogenesis, glycogenesis, glycogenolysis, and then there's this pentose phosphate pathway that kind of doesn't have any of the glyco in it because you're going from glyco to a 5-membered ring. So that's where the pentose comes in. All right, so I just wanted to give you kind of an overview. Again, sometimes I think that just putting a lot of words and arrows on a page can kind of help to solidify things. And sometimes just kind of going through the process and remembering how they're all created and connected helps as well. So I hope this helped you. If you have any questions, don't hesitate to reach out. Otherwise, I'll talk to you again soon.